Welcome, everyone. I'm Bill Sanders, the Dr. William D. and Nancy W. Strecker Dean of the College of Engineering. Welcome to the College of Engineering's webinar series, Faculty Insights, a 20-minute briefing. Thank you for being here today. In this series, faculty from across the College of Engineering share insights into their research, its impact, and provide a perspective for the future. Following each 20-minute presentation, attendees will have an opportunity to direct questions and comments to the speaker. Our speaker today is Professor Tzahi Cohen-Karni, who will share recent breakthroughs that his research team has achieved through the design and fabrication of engineering systems to tackle some of the biggest challenges, from supporting living pharmacies to helping wounds heal. Tzahi is a professor of biomedical engineering and material science and engineering at Carnegie Mellon. His team's research efforts have been focused on three major thrusts: synthesis and in-depth mechanistic investigation of the unique emergent optical, thermal, electrical, and electrochemical properties of novel hybrid nanomaterials and nanomaterial topologies composed on one-dimensional and two-dimensional building blocks. Application and characterization of hybrid nanomaterials interfaces with cells and tissue, and the development and engineering of nanomaterials-based platforms to integrate and affect the electrical properties of tissue and cells, such as cardiomyocytes and neurons, with the specific goal to understand electrical signal transduction in complex 3D cellular assemblies. Tzahi and his group strive to answer important questions such as, can material and platforms be tailored to allow seamless and stable integration with cells and tissue, as well as enable remote sensing and actuation? Can hybrid nanomaterials allow new insights into biological processes, such as tissue development and disease progression? Tzahi has received multiple awards, including the 2012 International Union of Pure and Applied Chemistry Young Chemists Award, the 2014 Charles Kaufman Foundation Young Investigator Research Award, the 2017 Cellular and Molecular Bioengineering Rising Star Award, the 2017 Office of Naval Research Young Investigator Award, and the 2018 Cellular and Molecular Biology Young Innovator Award. Before I turn the screen over to him, I'd like to remind our audience today to please use the question and answer feature found at the bottom of your screen to submit your questions to Tzahi during his presentation. You may do this during any point of the program. Nikki Bernardi, Senior Director of Development for the College of Engineering, will be moderating a question and answer session with Tzahi at the conclusion of his presentation. Tzahi, thank you for being here today. Over to you.
Bill, thank you very much for the opportunity to present my team's work to this amazing group of attendees. Let me share my slides and I'll start rolling, I say. Okay, ready to roll. So I'm going to talk today about tackling grand biomedical challenges with bioelectronics. Now, I decided to limit the discussion to one section of bioelectronics, so you'll see it in a bit. Just some disclosures at the bottom about startups that started out of the research we do here in CMU and with collaborators. A little bit about myself. I was born and raised in Israel, south to Tel Aviv. Did my undergraduate degree at the Technion in material science, engineering, and chemistry. I have master's degree in chemistry as well. And sometime in early 2000, I moved from Israel all the way to a small school that is not as good as CMU, Harvard University, where I obtained my PhD in applied physics. In 2014, I moved to Pittsburgh to start my independent career, where I instruct a merry bunch of PhDs, postdocs, and master students and undergraduates. As Dean Sanders mentioned, I am deeply interested in cooking materials and using these materials to dig deep in biological challenges. Most of my team, almost all of the students in my team learn how to cook. So if you join my team, you start with cooking either 0D, 1D, 2D materials, characterize them. Then some people are interested in using these materials to dig deep in understanding some electrophysiological properties of tissue. So this is an extensive effort in my lab, trying to elucidate how disease is altering, for example, electrophysiology of cardiomyocytes. These are the cells that build our hearts, basically, one cell type. Some students in my team are deeply interested in using unique optical properties of materials to change or modulate electrical activity of cells. So this is what you see here, basically shoot lasers on materials and alter the electrophysiology of neurons, for example. Some of the materials that we make or create in the lab have unique chemical properties, and we use them for sensing or basically catalysis. So within all this effort that we work on, I'm going to talk about mostly catalysis and its actually applications in bioengineering. So before I even delve deep into the topic I want to cover today, I should mention the people behind the hard work. Currently, I have 14 PhD students in my lab, a couple of 2 master's students and 2 postdocs working with me. The ones that appear as weird AI-generated image are just literally joined the lab a couple of weeks ago. So we still work on their images, basically. The team itself is across, you can see across almost all the engineering departments we have, Matsai, BME, MECI, that we have over here. The topic I decided to talk today is not everything on the original title, so I'm sorry about that. But I want to tell you about something that is new. Some research field that started 20 years ago, and this is biohybrid devices. So biohybrid devices, if I need to explain it to someone that is not from my field, is actually taking living matter and fusing it with digital world, or let's say electronics. The living matter are engineered cells that can produce therapy. And the therapy is any peptide that can be used to treat a disease. This could be peptides to treat cancer, peptides to treat diabetes, peptide to treat obesity, and any chronic state disease. Now, this is the bio end of the program, and I'm basically, this is done through extensive collaborations with colleagues across the country. The electronic end, or these devices that we work on, have a really unique capability when it comes to these biohybrid devices. They can sense a state of disease, or they can sense how much therapy was produced. They can actually actuate by sending in signals to stimulate cells to produce therapy. And at the same time, we have unique devices that can maintain these cells. So this type of therapies of approaches been deployed in multiple projects that I've been fortunate to work on in the last 5 years. The first project we started working on is actually bioelectronics for tissue regeneration. This is a massive effort under DARPA, Defense Advanced Research Project Agency. These are the agency that does all the WACO research in the US and outside of the US, such as developing the GPS or the internet, basically. Other efforts under DARPA is the N-Train and the TOR project is under ARPA-H, which is another agency toward healthcare or toward health. So all these ideas related to these implanted devices, so these devices could be as small as this thing that I'm holding in my hands, and later on I'll zoom on it using a different camera so you can see it. This device is basically less than 2 inches long or 4 centimeters long, give or take, and 1.5 centimeters wide. So let's say a bit more than half an inch, let's say. Okay, so I want to talk about something that my team been working on in the last 4 years or so, which is related to the maintenance of these implanted cells. And I want to give a quick tutorial about the importance of oxygen and why maintenance, as in providing oxygen to implanted cells, is what is driving now the technology. And just to give you an idea, this type of things I show you now are in the process of pushing them to the clinic. So that's the reason I chose this topic, because it's by far the most translatable effort I've been involved in the last many years. So in your body, in my body, any living organism, a mammalian, let's say, organism, any cell is within 100 microns from a capillary, blood capillary. And the reason behind it is actually the need for oxygen. So once the cell is getting far away from this capillary, it is under a condition that is called hypoxia, which means low oxygen concentration. And if in the last 30 years, scientists try to develop approaches to alleviate this or allow devices with living pharmacies, it's called, to sustain inside the body. But if you look at something that is called just diffusion-based devices, these devices are enormous because they need a large surface area to allow oxygen to diffuse into the device. So this is not, this is actually drawn to scale, this panel A on top right. So it's almost 80 centimeters long device to allow diffusion of oxygen to this device to maintain, in this case, pancreatic islet. So the research that this team was involved in was essentially treating type 1 diabetes. So this actually inspired scientists, as I said, to design smaller devices that can be implanted. So think about devices that will be the scale of a pacemaker or something along these lines. So few centimeters by few centimeters. But to do so, you need to have active production of oxygen or active supplemental oxygen, actively supplement oxygen to this device, as this particular illustration at the bottom right shows. That's the idea behind supplementing or providing oxygen and its need. So in my team, there are a bunch of people and collaborators at the bottom right. We are working on actually this type of effort, and we are trying to sustain these living pharmacies. So living cells that produce therapies. So this is not science fiction. This is the reality, I have to admit. Again, as I said, this effort didn't start from my lab. It started a long while ago by colleagues in Boston, actually, in MIT. So you have these immune isolation devices. These are devices that can have transplanted cells and an immune isolation membrane. And these devices are geared toward, again, having these foreign cells inside the body provide therapy. But as I mentioned, when you implant these devices in the body, let's say in a typical area such as a subcutaneous or under the skin, they're under a reduced oxygen tension, not enough oxygen, basically. Clearly, if I give it another visual way, if you don't have oxygen, basically these cells are dead. So red is bad, green is good. So red means dead cells. This is only after 48 hours under hypoxic conditions. Sometimes scientists try to resolve this issue by recruiting blood vessels blood capillaries to grow near these implanted devices, or some scientists try to deal with fouling, as in this capsule of proteins that will deposit on the device and actually serve as a barrier for oxygen to diffuse in. So these are the techniques that work really well in small animal models, but are not scalable to human beings. So where you will need probably a larger surface area to allow passive diffusion of oxygen. So again, we need to produce oxygen or provide oxygen actively. A few techniques that are out there, one of them is actually providing oxygen by using oxygen-producing chemicals. These are called peroxides, or calcium peroxide, metal peroxide, basically. The calcium peroxide will decompose to hydrogen peroxide that immediately will go toward the oxygen. The key advantage in this one is that you can provide oxygen beyond the concentration that the passive diffusion or motion of oxygen within our body. However, you have a depot of a material, a powder-like material that will decompose in a course of several weeks, up to 3 weeks, let's say, and you do not have control. So once it breaks down, it will diffuse into the nearby tissue and oxygenate, but you can't tune it. You can't increase or decrease. It's a fixed rate, basically. And it will be depleted. Another way, and this is actually was translated to a clinical trial through a company that is called Beta O2. And this one is actually using gases, oxygen, and it's fairly bulky device. You can see the size of the device. The scale bar is over here, where oxygen is literally injected into a chamber and then into a device that is implanted subcutaneous. So this is, of course, prone to infection. It's a bulky, massive device. In the last 10 years or so, there's a surge, a bit more than 10 years, there's a surge to develop an alternative approach, which is water splitting using electrochemistry. So these are devices that the beginning of this thrust is late 90s, some ideas, and then another wave of devices somewhere in let's say 2012, 2015, give or take, more papers about that. However, this approach did not use material science. That's actually why what we are doing is so unique. And this approach was again bulky and was not attempted in an animal model. So to show that this can be translated. So what we are bringing into the game is actually a bit different. We are taking all our knowledge of material science and deploying it into bioengineering. And I'll show exactly what I mean in the next slide. What we are doing is what probably a bunch of us did as young students some time ago by basically splitting water, electrolysis. You have anode, cathode, you apply potential across, you'll have oxygen at the anode and hydrogen at the cathode. But we are doing this chemistry in a smart way, unlike previous attempts. We are doing it using or digging deep into known information out there about how to make this process more efficient, as in lower energy that is needed. And water splitting is an uphill reaction. So you need to provide energy to get the oxygen that we are interested in. But we don't want to provide a lot of energy. So we want to reduce this energetic cost by using something that is called a catalyst or electrocatalyst. And these are the materials that we are utilizing in our own independent research. And I'll show it in a bit. So just to show you the inspiration, and which is ended up being this device that I'm holding in my hand, we will have this subcutaneous device that will be remotely powered by a battery, like in this case, a lithium-ion battery. We are using a catalyst, which I'll show in the next slide, which is based on a transition metal oxide. And we will have a special design that will allow us to control the way oxygen is being generated, and including the profile, the concentration profile. And we can turn it on and off or crank it up or down easily. And just to show you how we started the whole process is first we scanned through a lot of catalysts, and nowadays you can do it a lot thoroughly, I should say, using AI machine learning tools. But when we started this effort, the AI machine learning tools were graduate students that scanned literature and found best catalyst to use. So these are ruthenium or iridium based. We synthesize them here using tools in the nanofab and generate these nanoscopic catalytic high surface area materials that are called sputtered iridium oxide films. And we basically, with the help of smart undergraduate students, we designed an array that we can control the gradient of oxygen just by changing the way the catalyst is patterned. So all these black holes are basically syrup. Syrup is optically, you see it's black. So an undergraduate student, Kemi, this is Ian Jimino, is now in Germany in a fellowship, help us design the array and actually monitor how we can get a uniform flow of oxygen. You know, this is just one example of an electrode. So if you ask yourself, how the heck does it look? I don't have a scale bar, but the length of this chip is on the order of 20 millimeter. The weight of this chip is a little bit more than 1 centimeter. So it's around 20 by 10, let's say, millimeters. And you can see 8 arrays on this chip, basically, 8 oxygenators. And we checked the safety of this material. We did all the electrochemical characterization, and we know that we have around 300 millivolts of safe operational window where we can generate sufficient oxygen to provide to the cells. So you can see that the top diagram, above 1.9 volt, at the potential of 1.9 volt, we start producing some chlorine, and chlorine will evolve or will change into bleach. So we don't want it. It will kill our cells. We can actually fine-tune the amount of oxygen we have in our devices. We can either by changing the potential or by changing the duty cycle of the device. So let me show you some applications of this. So I think the first demonstration is in vitro. So we do it on a dish, basically, experiment that is done inside an incubator under 1% oxygen. So it's hypoxia condition. So to walk you through these visuals, the bottom part is basically a normoxia. So this is what we close enough to what we inhale in ambient conditions. So you can see that, again, green is good, red is bad. You see that there are many cells that are alive, but there are many cells that are not alive after 21 days inside this condition. When you take the same capsules, and I'll explain about these capsules in a bit, you take these capsules that are packed with cells, and you put them inside hypoxia, almost all the cells die after 21 days. When you turn on the switch and now the oxygenator is producing oxygen, almost all the cells are alive. So these capsules, by the way, are just a biomaterial that is mixed with living cells. These are cells that are engineered by a colleague in Rice University. We have fairly high density of cells in that capsule. We have something on the order of 60,000 cells per cubic millimeter, or 60 million cells per ml or milliliter. So that's a lot, basically. If I take these images and now translate it to a graphical way, you can see that our gizmo, this engineered platform, can sustain the cells up to 21 days. And I have one slide to show you way more than that. And at the same time, if you don't have the switch turned on, the cells just die. And what we also did to show production as a proof of concept, production of therapy. So leptin is a peptide, is actually controlling appetite, controlling a circadian rhythm. So our biological clock, which was part of the DARPA project I'm still working on. So we showed that with this gizmo that we made, these cells keep producing the peptide at the same rate. And once you don't have it, when you turn off the switch and it's not working, the cells just do not produce, mainly because they're just dead. We also developed really cool techniques, imaging techniques in the lab to show that via optical microscopy, laser scanning, confocal microscopy, to show that cells that have oxygenation have green cells throughout the capsule. So if you look at the capsule from the center of the capsule all the way to outward, basically, all the cells are more than 80% alive, give or take. The dead cells are just the capsules that do not have oxygenation. Most of the cells are dead. You have some handful of cells at the outskirts where there's some oxygen that can diffuse into these cells. So this is what you see on the right-hand side analysis. Let me show you something really interesting that we've done, that we actually push the limits of what's possible out there. And we basically increase the concentration, increase the loading of the cells to 8 million cells. This is by far the largest values known in literature. And now you have cells that are alive for a month in vitro. And after a month, all the cells in the control are dead. And our cells are just, the oxygenation magic is working, and more than 80% of the cells are alive. So this is a clear demonstration of maintenance and why you need it. Without it, any long-term device will not sustain. And the other thing that we did is push this technology into an animal model. So what you see over here is a different version of the device I showed you that is tethered. And now this device is residing in the abdominal cavity of this rodent. And we let it run for 10 days. After 10 days, we extracted these devices and characterized the state of the capsules, which is a biomaterial, again, mixed with a tiny sphere of biomaterials mixed with cells, with a density of 60 million cells per milliliter. And you can see that this type of approach allowed us to sustain the cells for 10 days with close to 80% viability, whereas without, when the switch is not on, these cells are dead. I have a lot more data to show in different animal models, etc., showing that this is working more than 10 days. We have efforts that I'm working on that is pushing it for 60 days and even more than that. But just to give you the flavor of these biohybrid devices and what they can be, what they can do. But you know, biohybrid devices are not just the maintenance. So what I want to show you is to give you the feel of what the future will be. Future is within the next few months, years to come, to be honest. So again, these biohybrid devices, this is the first slide I mentioned, is the mix of bioelectronics and cells, living pharmacies. And these cells are engineered to do something. The something could be treat a disease or release a certain molecule that can be used for diagnostic in response to a signal, for example. The bioelectronics is all these three blocks at the bottom. So we have all these electrical, electrochemical, and optical interfaces that can affect or supplement the cells, stimulate the cells, measure a certain analyte, and then stimulate the cells. So there are way too many kind of combinations of things that one can do associated with reading and writing information into the biological entities. I think just to give the vision for the next several years, what we will do is probably start dealing with diseases. So from this Lego set, we want to build parts that will allow us to treat diseases, chronic or acute diseases. Then that's what we are tasked with in let's say, these projects I mentioned. So I'll end up with that. What I want to also mention is No, actually, I'm done with mentioning anything. I am deeply thankful for the team I'm working on with. They are on this right and left-hand side, sorry. This is how we looked a bit more than 4 years ago. This is our cooking facility in Roberts Engineering Hall, first floor. This is us a couple of years ago, just to show you evolution of the group in a way. Now we are a lot larger than this. This is us last year. And this is us a month ago, actually, a month and a half ago, going to a kayaking trip an hour away. And of course, all this work is possible because of some agencies believe in us and know that we can do wild things here in CMU. And that's about it, I say. Questions, I guess.
Yes, thank you, Tzahi. We very much appreciate all of these great insights that you've shared with us today. We will now turn to our audience for questions. As a reminder, please submit your questions via the Q&A feature at the bottom of your screen. Make sure to include your name with your question. So before we get into questions, I just wanted to share a very nice comment we got from Joe Ciota. And I apologize, Joe, if I'm mispronouncing your last name, but he's a 1970s PhD MSc alumnus, and he's been blessed by the multidisciplinary mantra in his career. But back then, biochemists were not part of teams. So he's absolutely fascinated by how we have grown the CMU knowledge base and how it will proliferate for many years to come. Thank you. He just loves continuing to learn from you and others who share your great work. So, Tzahi, the first question I will pose to you is, what motivated the research you presented here today?
Oh, this is outstanding. I think personally, my motivation for many years since I started my own graduate studies was to fuse electronics with cells. And when I had these meetings with colleagues almost 6 years ago, that was clear that this is the way to do it, as in take what Omid Weisse in Rice University is doing and take all the goodness that we do here in CMU, fuse them together. So making cyborgs, this is what I did during my PhD, but making cyborgs, let's say, for a good cause, treat a disease.
That's wonderful. So this next question is from Raj. How can a CMU sophomore undergraduate chemie student get involved in research at your lab?
Send me an email. I have a bunch of them working with me. I didn't put all their images. I didn't put all the images of undergraduates over here. Nothing against them. Just email me, and I'm working with a bunch of them already.
That's great. So this question is from Leaf. How does hydrogen from electrolysis need to be removed before injecting the oxygen to the cell?
Oh, this is outstanding question. So all animals used are safe. No animal started floating around because of hydrogen. Hydrogen is actually fast diffusing, and it's being exhaled. The hydrogen we are making is in a very low concentration as well, by the way. It's a good point.
Wonderful. So what is the prospect of translating these efforts out of your lab?
It's it's outstanding prospect. I can't disclose too many details, but we have a program in last stages of approval to go first in human clinical trial very soon. So that's the reason I picked on this, because I want to show how applied engineering is making a big stamp out there, I have to admit. So this is by far one of the most advanced efforts. I don't know if it answered.
Yeah. Well, and this question is very similar, I think, than the one that was just posed. And where do you see the future of this work going, and what's the next big thing?
Whoa, that's interesting. So the future will be, in my mind, is basically having a way to treat multiple diseases using a single device, that I'll just kick this one so you can see, a single device that as small as this thing I'm holding, and I can, let me do something like this so you can see it to some extent. I don't know if you can see it. You can see it now, I assume, yeah? So I think a single device that will look like that and will be able to use multiple medicines in it and treat it and treat them, and the physician will be informed remotely. This will be like, you don't need to go to the doctor office to get your medicine. You don't need to take the medicine. Everything will be done by a smart device, I call it. That's how I think this will be the future, and I know for a fact that I'll be busy with that in the next several years.
Yes, I think so. Okay, so this question is from Matt Sklar. Is there any work being done in the area of muscular activation with electronic stimulus?
Oh yeah. Yes, we are working on that. Unfortunately, not unfortunately, I have to admit, I decided to focus on these biohybrid devices, but we have efforts in my lab and other labs in CMU related to skeletal muscle interfaces, how to basically stimulate, regenerate, control. We do that, and others in CMU do that as well.
So this next question, Tzahi, I think you talked a little bit about when it was asked about a chemi sophomore, but are there research projects that new CMU students can participate in? And I think by new, perhaps that means first year. And if yes, then what is the application process?
That's another great question. Ian Jimino that worked with me, Gaurav Balakrishnan that worked with me, Mabel Bartlett, she's a PhD now in my lab, and she started working with me as a freshman. Like basically, I don't mind what stage a student is, just join the team and start working with us. The way to do it is send an email.
There we go. Okay, so we have two questions from Raj. So the first is, how are you using AI in designing new nanomaterials or chemical processes to improve products?
I personally do not, this is one hat I don't want even to put on my head because I'm out of this field. I collaborate with people that's what they do. For example, Amir Baharati in our school, that's what he does. So I collaborate with him. So I work with people that know how to do it. Okay. Unfortunately, I'm not an expert in that.
Well, we can't be an expert in everything.
I guess so.
Okay, so the other question that Raj has is, can you use your technology to provide dopamine for Parkinson's patients?
Oh, that's a good question. I know that this is a question Omid Weiss, the scientist that is doing the cell engineering, was asked. And I think the answer is that there's a way to engineer certain cells to do it. So it won't be the traditional way I showed. It will be a different type of cells that need to be used. But I don't want to oversell. I am, again, not an expert in the cell engineering end or the synthetic biology end, but I think there is a way to do it, as far as I'm aware of.
Okay. This question is from Sharath. are students and PhD, are PhD students and other students allowed to apply for patents if they are involved in research at Carnegie Mellon?
Yes, my lab, to be honest, all the IP is not only my name, it's all the people that were part of the idea development. So it's always PhD or postdocs on this IP that we filed.
Okay. And another attendee asked, how can we support your work?
Oh, this is, I don't know if, Bill, if I should answer that. It depends
Of
what
course,
type of...
of course you should.
So there are some wilder ideas that are on the verge of being commercialized, and it's always a challenge, as you know, taking things out of the lab. And I'm fully open to have these conversations to talk about translation and where we stand in translation and what is needed to push things out from the lab to the clinic. This is like one aspect of the work. The other aspect is how can we have even better infrastructure within what we have over here, which is already amazing. But how can we have an edge over the rest of the competition? And that's a different discussion. So there's a discussion within the institute or within the organization I work with, and a discussion that is outside the organization. How can I take all the amazing things we do and kick it out? I'm open to have discussions, and I'll let the school manage this, I say. I'm just a mad scientist, kind of.
Okay, so we've had a couple of questions about this, so I just want to let everyone know that yes, we will be sharing the presentation and slide deck with everyone after the program is over. And Tzahi, I have one last question for you. What are the resources that enable these projects?
Okay, this is a great question. So usually the whole effort starts, these wilder ideas are funded by the federal government. As you all know, I mentioned DARPA. ARPA-H is another funding agency that is taking these moonshots or Mars shot, whatever you want to call them, and put lots of money to allow us to do it. We do have some of these wilder ideas started with seed funding through the school that helped us develop initial stages as well. But most of the work I showed today is funded through federal grants.
Wonderful. All right. Well, if there are no other questions, that will wrap up the programming for today. Thank you again, Tzahi, for a very thought-provoking and informative program. I would also like to thank our audience for participating today. I hope you will all be able to join us at future Faculty Insights programs. Please keep an eye out for a follow-up email soon, which will include a link to the recording from this session. And have a wonderful day.